Hey, welcome and good afternoon, everybody. We have a few more people rolling in, but we'll maybe get started with introductions and any kind of housekeeping. So this is, as you probably all know, the monthly Coregonine seminar. So it's basically a seminar series related to all things Coregonine for all the Coregonine enthusiasts out there. So this month we have Randy Brown giving us a talk. He is a fish biologist with the U.S. Fish and Wildlife Service in Fairbanks, Alaska. He earned his BS in biology and his master's in fishery science from Alaska Fairbanks, and has recently been given an honorary doctorate of science by that same university for his contributions to Alaskan fisheries over the years. So he's been working for the last 25 years on a wide range of investigations in northern Alaska, many dealing with Coregonine species that are harvested in subsistence fisheries. So what he's going to talk about today, so we're going to actually, for this week, we're going to shift our focus a little bit west and quite far north from the Great Lakes. And he is going to talk about a set of research efforts that he's been involved in to look at, to try and gain a better understanding of the biology and spawning behavior of Bering Cisco in the Yukon River. So I'll turn it over to Randy at this point.
Okay, thank you, Wendy. Let me get the presentation up. And see if we can get going here.
And just while Randy is getting ready, just a reminder to folks to check and make sure your mic is off.
Okay. I assume everybody can see this and hear me, but yeah, so thanks for coming today. I'll be speaking here on some Bering Cisco migration research I've been involved with over the last 25 years that has fundamentally expanded our understanding of this extraordinary species. Bering Cisco is an anadromous North American Coregonid species with a terminal mouth and non-pigmented ventral fins. The species appears to be endemic to Alaska and adjacent waters of the Yukon River in Canada. I'll discuss a number of research efforts that have taken place since the mid-1990s. Some were simply opportunities that arose while engaged in other projects, while others were directed to specific aspects of Bering Cisco biology. These include migration timing, demographics in the Yukon River, genetics, baseline development, and mixed stock analysis, otolith chemistry work to identify migrations to sea, and coastal distribution, radiotelemetry work to identify spawning habitats, population-specific morphology work, and mark recapture project, and a mark recapture project in the Yukon River to estimate the magnitude of the spawning migration. It should also be noted that the Office of Subsistence Management, the Fishery Resource Monitoring Program, provided major funding for several of these research efforts. There are numerous additional people who've contributed in one form or another to these research efforts as well, too many to list individually. I was first introduced to Bering Cisco while working on a chum salmon tagging operation on the Yukon River in 1996 at a place called Rapids, about 1,176 kilometers upstream from the sea. The U.S. Fish and Wildlife Service had contracted with a local fisherman, Stan Zaray, to run his fish wheel as a capture operation for us. Rapids is a narrow reach of the river that flows reasonably fast. Fish migrating upstream tend to run close to shore in the area, making fish wheels very effective. Our target species at the time was fall chum salmon, but the fish wheel was catching a wide assortment of fish, including 3 Pacific salmon species, 5 Coregonus species, and an assortment of others. The fall chum salmon tagging project lasted for 10 years. Early on, there was an effort to automate the fish recapture operation so fish wouldn't have to be held in a live box and dipped out with a net to see what the total catch was and how many tagged fish were there. Stan worked with Tevis Underwood and Dave Daum with the U.S. Fish and Wildlife Service to set up a video system on the fish wheel to capture high-resolution images of each captured fish. Dave published a paper on this technology in 2005. One of the unexpected benefits of this was that the collection of relative abundance data for all species migrating along the river. Stan's fish wheel eventually became a mainstem fish monitoring program that ran for almost 20 years altogether, from early June through late September. Stan received substantial funding from the Yukon panel for this work. Relative abundance data for Bering Cisco were collected from 2001 through 2015. Bering Cisco were present every year in June when the fish wheels began turning. Had 2 or more major spikes in abundance over the summer and tapered off in September. The maximum daily catch during this period exceeded 700 fish during a strong pulse in 2010.
Sorry, Randy, I accidentally muted you there. Could you unmute?
Unmute. There we go. Otherwise, going good?
Yep, all good. Sorry, I was admitting somebody and I hit
Okay.
mute by accident.
Okay. So in this slide, you can see differences in temporal migration patterns between Bering Cisco and some of the other Coregonus species. And it's a 7-year selection, so we can see it, but the annual CPUE data from for Inconnu and broadwater fish, for example, are low during most of the summer and rise significantly during late summer and fall, while Bering Cisco follow a more irregular pattern of migration. Uh-oh. There we go. This plot included the cumulative CPUE values for the years 2001 to 2007, and it illustrates the large differences in relative abundance between the Bering Cisco runs and those of the other Coregonid species. In 2010, the cumulative CPUE for Bering Cisco was over 10,000 fish. So are you folks seeing this bar at the top? The sharing bar and everything? I hope not.
No, I don't think so.
Okay. In 2005, an experimental commercial fishery for Coregonid species began in the Yukon Delta. By 2007, the buyer in New York City wanted Bering Cisco, but not the other species. This 2008 New York Times article and a couple of others after it featured the smoked Bering Cisco from the Yukon River as a kosher delicacy. These fish were substituting for a deep water Cisco from the Great Lakes, referred to as chubs, that were apparently no longer available. The fishery was taking place in the lower reaches of the south and north mouths of the Yukon River and nearby coastal areas along the delta. Meanwhile, several of us working at Rapids have been doing work documenting the fact that the Coregonid fishes migrating past were on a spawning migration. I presented the work we'd been doing at a Coregonid conference in Winnipeg, and we published a paper in the proceedings. This is a figure from the paper illustrating the significant rise in the gonatosomatic indices over the summer for all of the Coregonid species. Even in the early 2000s, it was unclear whether all of the Coregonid species we were seeing at the Rapids were anadromous or not. I had done some otolith chemistry work with Inconnu and showed that the group migrating past Rapids was overwhelmingly anadromous, and my colleague Bill Carter was doing similar work with broadwhitefish. This paper addressed all of the species at Rapids and expanded to Coregonids in other areas of the drainage as well. I conducted sampling programs for Coregonid species in a number of locations and persuaded a few of my colleagues with ADF&G, as the Alaska Department of Fish and Game, and other rural fishers I knew to provide additional samples. All in all, I was able to compile collections from 8 sites in the drainage as indicated by the arrows with numbers here. So I had information on species, collection locations, lengths, weights, gonadosmatic indices of females, and otoliths from all the fish collected. The chemistry component of the investigation required taking a transverse section of an otolith from each fish through the core as illustrated here, mount it to a glass slide, and view it with a compound microscope to age the fish. This is an image of an old Bering Cisco that we aged at 13 years. The groove on the top is the sulcus and the core is the dark area below the sulcus. This is an otolith of an age 6 Bering Cisco with a transect for aging and microchemical analyses illustrated from the core to the margin across all the growth increments. The electron microprobe uses an electron beam to sample a series of spots along a core to margin transect and collect quantitative data on elements of interest from each one. These spots are 5 microns in diameter and 8 microns center to center. To identify anadromy in fishes that are not obligated to migrate to sea, we sample for strontium and calcium concentrations. Otoliths are calcium carbonate structures that will incorporate trace elements like strontium in proportion to its abundance relative to dissolved calcium in ambient water. Most fresh waters have low ratios of strontium and calcium, much lower than the marine water at about 8.6 millimoles per mole. This is a figure illustrating this phenomenon from a paper that Ken Severin and I published in 2009, illustrating the distribution of the ratio of strontium to calcium among world rivers, lakes, and oceans. The strontium-calcium ratio for the Yukon River is indicated at about 1.9 millimoles per mole. Here's the strontium-calcium profiles from 3 coregonid fish with known life histories, illustrating the significant rise in strontium-calcium in the 2 fish known to be anadromous. Bering Cisco were only found along the main stem of the Yukon River, not in any of the tributaries where we sampled, and all Bering Cisco were found to be anadromous. Here are 4 examples of strontium and calcium profiles illustrating what we think are slightly different life histories. Like other salmonids, Bering Cisco can't survive in the sub-zero water temperatures under sea ice, so they need to withdraw into brackish water estuaries or the lower reaches of rivers for winter. This phenomenon can be observed as regular dips in the strontium and calcium ratios in the top right and 2 bottom profiles. The top left profile appears to show an extended period of purely marine residency, suggesting that individual may have been in the southern Bering Sea or Alaska Peninsula where the sea wasn't frozen. And then this figure illustrates our findings for all the species on the upstream extent of anadromous coregonid migrations for the species we found. The lower left panel shows that Bering Cisco were found to be anadromous throughout the Yukon River main stem, all the way to the border with Canada, which is a distance of about 2,000 kilometers. Other species found upstream from the Yukon Flats in the Porcupine and Yukon rivers were not anadromous. Additionally, all the Bering Cisco we found in fresh water were preparing to spawn, with females carrying large egg skeins, and none were feeding. Meanwhile, some involved with the commercial fishery in the Yukon Delta believed that the fish they were harvesting spawned in the Delta and didn't migrate upstream. Those of us working on Bering Cisco in the upper river were asked to comment on this, and we suggested that the management team collect gonadosmatic index data and compare to our data from rapids and other upstream sites. This figure illustrates the samples collected from the commercial fishery in fall and winter, black circles near and below the GSI = 3 reference line, compared to those from upstream reaches in the drainage, which are the blue circles following the blue curved line, to levels as high as GSI = 40% in September and October. These data convinced everybody that the commercial fishery was harvesting non-spawning fish. My colleague Dave Daum and I have been scheming for a number of years to see if we could figure out where Bering Cisco were going to spawn. We did some exploratory work to see if we could successfully implant radio tags in them at rapids and have them continue migrating upstream. We found that they accepted the tags well, and we finally got funding from the Office of Subsistence Management for a large-scale 2-year effort to locate spawning habitats. This is one of the tags, small by necessity, but with enough battery power to transmit through the summer and fall. At the same time, we were working to find the Yukon River spawning area. Mike Thalhauser, a fish biologist with the Kuskokwim Native Association, got funding to do similar work with Kuskokwim River, Bering Cisco. He joined us on the Yukon River to gain experience doing surgeries. This is Mike, the anesthesiologist on the left, and me, the surgeon on the right, sewing up an incision on a Bering Cisco at rapids. We monitored the migrations of tagged Bering Cisco with remote receiving stations at critical locations along the river, so we knew how many fish were in various regions of the drainage. This station is mounted on a high bluff along the Yukon River about 10 kilometers upstream of the tagging site. We tagged them at rapids during a number of periods during 2012 and 13. We used 2 remote radio stations during the project, one upstream from rapids to identify those that recovered and continued migrating upstream, and another upstream from Circle to identify any fish that migrated beyond the Yukon Flats. By late September, about 80% of our radio tag sample migrated to the upper Yukon Flats. The other 20% were distributed in the Yukon Flats downstream from the mouth of the Porcupine River, and 3 fish migrated upstream into Canada for spawning season. During the fall of 2013, the project nearly fell apart when the US government went through a political shutdown from October 1 through 17, encompassing the entire spawning period for Bering Cisco. They clearly didn't consider this fact in Washington when they shut down the government at that time. However, I approached some of my colleagues with the Alaska Department of Fish and Game, and they ended up flying the spawning season surveys for us that year, which saved the project. This is a Google Earth image of the heavily braided reach of the upper Yukon Flats, where most of the Bering Cisco population migrated to spawn. It's a wild braided stretch of river. This is how the reach looks from an airplane flying a radio telemetry survey. It can be more than 5 kilometers wide from main bank to main bank, and this river morphology extends for about 125 kilometers. Mike Thalhauser found his radio tag fish in the South Fork Kuskokwim River, with many of them in this reach during late September and early October. The South Fork spawning habitat clearly had similar qualities to those of the Yukon Flats. During the early Sioux Hydro fishery investigations, the Alaska Department of Fish and Game had a number of fish wheels distributed along the main stem Susitna River and its larger tributaries. They found that Bering Cisco returning from the sea avoided the tributaries, migrated up the main stem to just shy of the confluence of the Chulitna, Susitna, and Talkeetna rivers. And they used electroshocking boats to locate concentrations of Bering Cisco in the fall and found them spawning in this braided reach in the vicinity of Montana Creek, just downstream from the community of Talkeetna. At this point, we had a solid understanding of the 3 spawning populations up the Yukon, South Fork Kuskokwim, and Susitna rivers, indicated as upstream shaded regions in this map, plus a single record in the Canadian community of Dawson. The marine distribution was originally described in an important paper by MacPhail in 1966, where he differentiated Bering from Arctic Cisco based primarily on differences in lower arch gillraker counts. MacPhail found that Bering Cisco were widely distributed in estuaries and coastal habitats of Western Alaska and in Cook Inlet. Since that time, 2 Bering Cisco were identified by Cherezhnev in 1981 at the mouth of the Chigutun River on the north side of the Chukotsk Peninsula. Miranda Plum captured one in the lower Ugashik River drainage on the north side of the Alaska Peninsula while she was conducting a fisheries investigation there. And Cheryl Anderson found some small numbers of Bering Cisco at a tidewater weir on a small stream in Cold Bay on the southwest tip of the Alaska Peninsula. Additionally, fisheries investigations associated with the Outer Continental Shelf projects in the 1970s and 1980s captured Bering Cisco in the middle of Norton Sound, which demonstrated that they aren't always close to shore in marine waters. In addition, they were found in coastal waters along the west side of Cook Inlet. Given this information, we considered the commercial fishery again. We figured the harvest could potentially be composed of fish from the Yukon and Kuskokwim rivers, but were unlikely to include fish from the Susitna River. So we discussed the idea of a genetics mixed stock approach to sorting out the population composition of the commercial harvest. We proposed that the U.S. Fish and Wildlife Service Conservation Genetics Lab take on the challenge, since they'd been working on a number of Coregonus projects already, and the Alaska Department of Fish and Game embraced the idea. Ora Russ took on the project for the U.S. Fish and Wildlife Service, and we began soliciting support for baseline samples. At the same time, Andy Padilla with the Alaska Department of Fish and Game Commercial Fisheries Division decided to approach the mixed stock issue from a strontium isotope chemistry perspective, in case the two Western Alaska populations were too similar to distinguish with genetics. If the Yukon and Kuskokwim rivers had distinctly different ratios of strontium-87 to strontium-86, then an otolith chemistry-based mixed stock assessment would be possible. Many people and organizations became involved in collecting population-specific genetic samples and otolith samples for these two research efforts. This is an extraordinary crew of ADF&G and U.S. Fish and Wildlife Service biologists collecting Bering Cisco from the spawning area in the Susitna River with the giant Alaska Range peaks in the background. On the genetics front, Ora obtained the baseline tissue samples for 3 spawning populations, 209 from the Susitna, 265 from the Kuskokwim, and 271 from the Yukon. She was able to establish baseline genetic qualities that clearly differentiated the 3 populations using 17 microsatellites. The Yukon and Kuskokwim River populations were more similar to each other than either was to the Susitna River population. This is a bar chart of the results of Ora's mixed stock analysis of 3 annual sample periods, with North and South Delta harvests broken out separately and sample sizes indicated at the top of each bar. Almost all of the fish harvested in the Yukon Delta fishery were of Yukon origin. This was an important finding for the fishery managers. On the otolith chemistry front, Andy Padilla had baseline samples from all 3 populations as well, 30 from the Susitna, 25 from the Kuskokwim, and 27 from the Yukon. Analyzing ratios of strontium-87 to strontium-86 to identify natal streams requires one to identify the section of the otolith that precipitated before the young fish left their natal habitat and migrated to sea. In addition, the very core of otoliths of anadromous species are often influenced by nutrient material from the egg with origins from the mother's foraging at sea. Strontium-87 to strontium-86 ratios in that region of an otolith will not be a reliable indicator of natal habitat. Therefore, a laser ablation transit crosses the otolith from one side of the first growth increment through the core and to the other side. At that point, its direction can be adjusted to cross all the growth increments out to the edge, as illustrated here. This results in 2 related plots. The top graph reflects the intensity of the strontium-88 signal, which is proportional to strontium elemental abundance. So the point at which migration to sea can be identified. The bottom graph includes all of the individual strontium-87 to strontium-86 ratio estimates, which are the circles, along with a low smoother, which is used to illustrate the best estimate of the ratio along the transect, essentially a running mean. Also included are the reference lines indicating the global marine value of strontium-87 to 86, which is GMV in this figure, and the value for the Yukon River at the bridge in Alaska. The bridge is just downstream of the Yukon Flats where they spawn. The two vertical dashed lines bracket the region of the first growth increment just to the right of the maternal peak in the very core of the otolith and just to the left of the distinct spike in strontium intensity that comes with the first migration to sea. The strontium-87 to strontium-86 ratios in this segment are used to identify the natal stream strontium-87 to strontium-86 ratio. This fish clearly had natal origins in the Yukon River. So the ratios of strontium-87 to 86 are essentially constant in marine environments around the world at 0.70918. The ratio varies somewhat among rivers because strontium-87 is produced through radioactive decay of rubidium-87, which has an extraordinarily long half-life. By contrast, the concentration of strontium-86 doesn't change over time. So rivers in drainages with very old rocks have more strontium-87 and higher strontium-87 to strontium-86 ratios than drainage basins with relatively young rocks. This plot includes reference lines for the strontium-87 to strontium-86 ratios for the Yukon, South Fork Kuskokwim, and Susitna rivers, as well as the global marine value. The box plots illustrate the spread of natal strontium-87 to strontium-86 ratios for the three baseline collections. The Yukon River has the largest spread of data among these three baseline samples, and we suspect that the much greater spawning area in the Yukon River leads to an increased likelihood of exposure to different water chemistries during early development. The otolith strontium isotope ratio analysis of stock composition of the commercial fishery showed similar results to those from the genetics mixed stock analysis. The harvest is composed of almost 98% Yukon origin fish. Once the fishery component of the project was complete, Andy pursued analysis of samples from coastal regions where the natal origins were not known. We collected otoliths from a number of sites in northern and western Alaska that are numbered in this map. Larry Moulton from Owl Ridge Consulting shared Bering Cisco otoliths from the western Beaufort Sea. Similar collections were provided by Bill Morris, who was with ADF&G at the time, for the northern Chukchi Sea. Samples from the Yukon Delta south of the Yukon River were provided by ADF&G and from Dave Runfola, who studied Bering Cisco in Scammon Bay for his master's degree. The one Bering Cisco from Miranda Plum, who was working in the lower Ugashig drainage on the north side of the Alaska Peninsula, was also available, as were 7 samples from Cold Bay that were provided by Cheryl Anderson on the southwest side of the Alaska Peninsula. In this sample of 126 Bering Cisco from coastal environments in western Alaska, 121 were clearly of Yukon origin and only 3 of Kuskokwim origin. Even the 7 fish from Cold Bay on the south side of the Alaska Peninsula were Yukon origin. Our tentative conclusion here was that the Yukon population is enormous compared to the Kuskokwim population. Notice that 2 fish were classified as Susitna origin, one collected from the Colville River Delta on the Beaufort Sea, and the other from the Coon River near Scammon Bay in the Yukon Delta. We actually think these are Yukon origin and that few, if any, Susitna fish migrate around to the north side of the Alaska Peninsula. In our original baseline samples, we had 1 fish collected in the Yukon River with a strontium-87 to strontium-86 ratio consistent with the Susitna population. However, remember that a single fish had been captured in Dawson in 1977, and that during the telemetry project, 3 of our tag fish had migrated upstream from the Yukon Flats and into Canadian waters during spawning season. 2 of them returned downstream past our station in late October, suggesting spawning upstream somewhere. We suspect that there is a spawning group of Bering Cisco in the Yukon River just downstream from the mouth of the White River. The White River has low strontium 87 to 86 ratio, very similar to the ratio in the Susitna River. This is an image of the confluence of the White River coming in from the left in the Yukon main stem, entering from the lower right, indicating that the stream morphology is similarly braided as in other Bering Cisco spawning areas. The developing eggs of Bering Cisco that spawn near the confluence on river left would experience White River chemistry, and on river right, main stem Yukon chemistry. The eggs of Bering Cisco spawning farther downstream would experience a greater mix of water chemistry from both forks of the river. We think that's what's going on with those low strontium 87 to 86 ratio fish from the Yukon. As we gathered baseline samples from the 3 spawning population, we realized that if we collected a number of whole fish samples, we could update McPhail's 1966 landmark taxonomy paper differentiating Bering Cisco and Arctic Cisco with population-specific morphological data. Several years after completing all the counting and measuring, Andres Lopez, who's a professor at the university here in town, in Fairbanks, and I finally published a paper on this work. Some things like the lateral line scale counts were similar among populations and similar to values from McPhail. Lower limb gill raker counts, however, were similar among the Western Alaska populations, but significantly different than the Susitna River population. Because McPhail's Bering Cisco samples were primarily from marine water, and we don't know how many Cook Inlet fish he captured, it may be that the slightly different distribution of values he obtained for Bering Cisco were simply because it was a mix of the 3 populations. The one final component of the research effort was the spawning population estimate. Once we had the spawning area identified, I met with some of the research staff at ADF&G Sportfish Division in Fairbanks, Matt Evenson, Klaus Wudick, and James Saverai. The Fisheries Resource Monitoring Program had recently put out a notice of funding opportunity with a priority information need related to Bering Cisco abundance in the Yukon River. I proposed to Sportfish folks that they could do a mark recapture project in the upper Yukon flats using electroshocking boats in the fall to tag fish and a couple weeks later to do a recapture event. Sportfish in Fairbanks specializes in mark recapture project, and I was confident that it was doable. Our telemetry data indicated that in mid to late September, the Bering Cisco were almost all present in the spawning grounds, and they wait around, moving very little until the 2nd week of October to spawn. Sportfish did an experimental run from Circle to see if they could capture large numbers of Bering Cisco in late fall, and that effort was successful. OSM ended up funding the 2-year project to estimate the spawning population of Bering Cisco in the upper Yukon flats, where about 80% of our radio tag fish went. The 125-kilometer reach was divided into 10 12.5-kilometer segments, which was a distance that 2 electroshocking boats could cover in a day. They worked their way downstream just offshore from gravel beaches, sandbars, and islands on both sides of the river. They would net fish for a period of time, put them in a live well tote behind them. Then they would take breaks to tag and release the captured fish, and then continued on each day for 10 days. They returned again a few days later to start over again at the upper part of the reach for the recapture event. These are the results from the 2 years of mark recapture work. Over 5,000 fish tagged in 2016 and over 4,500 in 2017. Over 8,000 and 10,000 fish were examined during the respective recapture events during the 2 years, with 105 and 131 of the recaptured fish being tagged. The spawning population in the upper Yukon flats was estimated to be a little more than 387,000 fish during both years. The actual spawning population is undoubtedly a bit more than this because not all spawning fish are in that geographic region. But these numbers provided some comfort to the commercial fishery managers that their limits on the fishery, 25,000 fish per year at the time, were appropriate and would not harm the population or impact the subsistence fishery. Details of the project, the statistics, and other related issues can be found in the ADF&G Fishery Data Series report that Matt Albert produced. During the course of this these research efforts, we learned a great deal about the biology of Bering Cisco. Mature females tend to be larger than mature males. Females tend to grow faster than males beginning early in life. These are von Berlanti growth curves fit to female and male length at age data. Females also tend to be heavier at length than males. And using age data from the commercial fishery and from the spawning population, we could see that the fishery captured fish that were about a year younger on average than the spawning population. Virtually all of the age 3 and age 4 fish from the commercial fishery are immature, and fish older than this could be a mix of immature and surviving mature fish. We always suspected that Bering Cisco suffered huge annual mortality, although we didn't know how much until we had a large amount of age data. We applied Robeson and Chapman's 1961 method to estimate survival based on the catch curve age data from the commercial fishery and from the spawning grounds, and achieved reasonably similar results. Our data suggests that a little less than half of the adult population survives from 1 year to the next. It should be clear to everybody that I think Bering Cisco is a fascinating species with an amazing life history, and hopefully in hearing the story, you share some of my enthusiasm. Thank you for tuning in.
Okay, thanks so much, Randy. That was a great presentation. So we have a little bit of time for questions. So folks can either use the chat to insert a question or put up their hand, and I'll try and see people as they put up their hand, and then also keep an eye on the chat.
Okay.
So Andrew has his hand up.
Yeah, cool talk, Randy. Really appreciate that. Just wondering, on the basis of the telemetry data, if you made any observations on behavior of the animals. So did you notice that females were moving in later than males into the river, or sooner, or anything like that, or staying longer on the spawning grounds?
Yeah, so we did get downstream, the later part of your question first, we did get downstream migration data from our telemetry towers, but it was real, since they're going down in late October, it was real fragmentary. It was just a few fish, which let us know that at least some of them leave, because they don't eat at all going upstream. And they look wasted on the spawning grounds when we've sampled there. So we were considering that it was possible that they all died, and but that doesn't happen. We've actually had some of the tagged fish from the population estimate be captured the next year down along the coast, along the west coast. So not very many, but some. So that means that some of them make it down, but we just don't have a rich enough data set on the passage. We get all of them that migrate upstream. We get them for about 2 hours migrating by these stations, and but we only get some of what goes downstream. So can't tell that. And we really don't know if there is a temporal difference. If it is, it's minor, or it'd be a proportional estimate that's not very different between males and females.
Great, thanks.
Yeah.
Okay. Thanks. Good job. Any other questions? Okay, Randy. You have to turn off your mic, Randy, or turn on your mic, Randy.
Okay, how's that?
Yeah, good.
When
thanks.
you did your morphometric study on Bering Cisco, did you consider size correcting your data, or did you not consider size correcting the data?
Well, we did not. We did it directly. And then there's, the small ones are right around 30, 32 centimeters fork length, and the big ones up towards 42, 45. And so, but we did not do a size correction. And that was really true for all the populations, you know, all three of them. I mean, they were, the Susitna were somewhat smaller and the Yukon somewhat bigger than the others, but it was not a significant, I mean, it was significant, but it wasn't a giant difference. So we didn't do size correction.
Well, we looked at your data and you're right. You did not need to do it.
Yeah.
So, and another question, do you have any morphometrics on in-canoe?
Well, no. It would be good to do that. We didn't work on that with in-canoe.
Okay, well, yeah, it's hard to find. Yeah.
Yeah, it is.
All right, thanks, Randy. And next up, Yu-Chan.
Hi, this is Chen Gao from Great Lakes Science Center. Thanks for your presentation. I think it's very interesting to learn your experience and to learn how the science and the knowledge accumulate over time. So my question is, when you started this, how do you decide what kind of research or what kind of knowledge you want to do first? And was there a point in your career you feel like, well, I wish I had known or I had done this first?
Yeah, so we really, you know, like I pointed out in the beginning, we started doing a chum salmon project, and these were bycatch, and there were, you know, the Coregonus were bycatch, but there had been, there was a fellow, Ken Alt, he was at Alaska Department of Fish and Game biologist that had worked for years studying these animals, but he was kind of, his career went through before radio telemetry became really effective in the Yukon. And he tried to deal with distribution by sampling, you know, and sampling works fine, but it's a giant river and it's big tributaries. And it was really challenging. He never did find the spawning area for those sheepish, and he was still ambivalent about whether, he thought they were anadromous, but he had not been able to put it together with definitive data of tagging down in the Delta and having them come up to the Yukon flats. And so when we were first catching these fish and realizing that we would catch, you know, 50 or 80 of these big in-canoe every day in the fall coming through there, along with Bering Cisco, lots more Bering Cisco and broad whitefish and humpback whitefish. And we were going, wow, this is really something. And some of the fish and game biologists said, you ought to use this opportunity to figure out where these fish are going to. We had a whole tower network into the upper drainage for the chum salmon project, because part of it was mark recapture, but part of it was telemetry. John Eiler with National Marine Fisheries Service was working on the telemetry component. So it was really opportunistic. The first step, you know, I did my master's degree there with in-canoe out of Rapids, and we did the biological work to find out, one, were they spawning? Were they all spawners? Because nobody really looked, you know, the fishermen that caught them didn't really look at that. They would put them up mostly for dog food. They're the great people food, but they also had salmon, and so they preferred the salmon mostly. They ate the in-canoe too, but they would put them up for dog food. But we found out they were all preparing to spawn, all of them. None of them were feeding. And so that means they're migrating somewhere. And I did otolith chemistry work with Ken Severin, who was at the University of Alaska with the electron microprobe work. And we found out that they're all anadromous. Okay, so they're coming from the ocean, they're feeding in the ocean, they're migrating up. Where are they going? And we added some of the tags that we were using for the chum salmon onto that. So we found this out with in-canoe for the Yukon main stem. And all of those in-canoe go into that same upper stretch of the Yukon flats, that same big braided area. Everybody thought, well, they're going to go up the Chandler River, they're going to go up the Porcupine, they're going to go up some other tributary they didn't go up the tributaries at all. They spawned right in the main stem Yukon River, big water. And so this led us to some of the other species that were also going, well, I'll bet they're just as interesting, and they are, and some of them even more so. And so we sort of stumbled upon it and just progressed in that way. And for Bering Cisco, the initiation of that commercial fishery really inspired the finances. The Office of Subsistence Management is a Fish and Wildlife Service funding effort that came about because of ANILCA, the Alaska National Interest Lands Conservation Act. And it's a federal program to provide funds for fishes that are subsistence resources and within the state and cross or live in federal conservation areas like parks or national parks or refuges. They provide millions of dollars on an annual basis to this work, and it's actually helped fund quite a bit of the Coregonus research, including most of the Bering Cisco stuff. So we had a funding source, a fishery that inspired some attention, and some background knowledge from rapids. But it wasn't like we thought all this stuff out beforehand. We just stumbled upon it, one thing after another as we went along. So, but you could use it to initiate a program for a species that nobody knows anything about, I think. I don't know if that.
Yeah, thank you.
On a ramble, but.
All right, thanks. And I see Ted has a question. Ted,
thanks, Randy. It was a great presentation. I just had a question. Maybe it was a relic of the tagging, but I think you said a big slug of fish would go by a receiver in about 2 hours. Does that mean, is that a relic of when they were tagged together and where they were tagged, or was it that they just move up as a whole pod? It seems like,
No. So what I was talking about was as soon as the fish, so that receiver that you saw, it's got antennas going both ways, upstream and downstream, and it covers, 2 or 3 miles downstream, 2 or 3 miles upstream. So they have 5 or more miles where they're migrating up, and Bering Cisco run real close to the shore and real close to the edge. And so from the time we would get a record of them at that station till the time they had passed and gone farther upstream, they would be in view of it for 2 hours, sometimes more.
Okay.
So we didn't miss any going upstream.
Right. Do they just kind of trickle up? Or do they come up as a giant pod in general? Like if you had tagged fish from different years?
No, they trickled up. They just, it was, so we would tag them, and usually they would sulk a little bit. It might be, 2 days or 5 days, and then they would start going upstream. And some of them would get recaptured or get captured in other fish wheels. Fish wheels are deadly for the Coregonus moving along the sides, but some of them would get got. And one that we let loose didn't want to dive. It stayed kind of close to the surface, and an eagle came and got it. So we had to watch for him. But but they would just move along, and we started doing aerial surveys in September, and they were all just sitting there waiting in the spawning area.
Okay.
And they would move a little bit, but not much. And we had some records from the lower tower and the upper tower, and they averaged around 13 kilometers a day for the trip up between those two towers. So.
Okay, thanks.
Thanks. And Andrew, and just a reminder, you can put your hand up or type something in the chat if you like.
Well, I don't know if people can see this, but I want to make a plug for Randy's report here. This is Alaska Fisheries Data Series number 2012-4, but this has got a lot of really cool information about the story of these whitefishes. So if people can get a hold of that, it's a pretty cool report. It's thick. There's lots in there. Randy, just one other question about the gill raker data you had shown there, showing that there were some differences between the McPhail distributions of gill raker counts. And I wonder if you looked into that further and had a sense of whether it really was just that he was sampling different fish than you guys, as opposed to changes in raker numbers over time, or population specific differences in raker numbers.
Well, I think that I don't have a way to know whether there was any change. I don't think so. I think that is pretty stable. What I think was happening, because most of his samples were taken along the coast, and he actually sampled in the Yukon as well for some of those samples, and then he sampled Cook Inlet. But I have no way of knowing how many. We looked through that paper a lot, and I don't know how many he got from the different areas, because he was going all around the Chukchi and the northern Bering coast. He went into Bristol Bay and caught some there, and then he went into Cook Inlet. And so I just think his did not have so much of the population specific aspect to it like we did. We got them off of spawning grounds, or as they were approaching spawning grounds. And so they were individual population data. And so, and given what we saw about the distribution, the population distribution along the Bering Sea and the Chukchi Sea from Andy Padilla's work there, I think most of his work in the marine system was Yukon, probably just a very few out of Kuskokwim, but those two were really identical. And then whatever he got from Cook Inlet, and we don't know how many, but I think it was just he just had all of them put together.
Okay, great. I don't see any more hands up. Can I ask another question, Wendy?
Yeah, go ahead. Actually, I was going to ask you, if you have it handy, can you drop a link to that report into the chat?
Yeah, I can look it up.
Yep.
Okay, so this may be an unfair question, but we're starting to see some river spawning Coregonians coming back in the Great Lakes. There's whitefish population, lake whitefish, that are coming back in Green Bay, for example. And there may be some river spawning Ciscos as well. And I guess my question is, knowing what you know about anadromous Coregonians on the coast, is there any sort of lessons you think you would want to transfer to us as we start to think more about the rebound of some of these river spawning Coregonians, or alternatively restoration of populations and habitats that once existed?
Well, that's getting out of my realm, but the river spawning fish, we've got a lot of data on that. So least Cisco, humpback whitefish, and round whitefish are capable of living in bound lakes. And so they must be spawning over gravel or rocks in those lakes, so it's not flowing water. All the others, we just find them in flowing water over gravel. The broad whitefish, the Bering Cisco, the Inkanu. And so I don't know. It's all different. We have the Bering Cisco. We're in these big braided systems. And the Susitna and the South Fork Kuskokwim were big glacial outwash rivers, right? And the Yukon Flats is not that. It's but it is a very steep. The water moves really fast through there. It's small gravel material, and I think what happens is there's a lot of water that flows underneath the islands and comes up. There's all these open leads in the lower ends of islands going through that whole thing, and I think that is really the reason why it's such a profoundly good destination for spawning, productive for Coregonan spawning in there. Similar habitat on the Tanana River, but then you get up to the Alatin River, and it's this big, meandering slow water stream, and the Salukna River which we've studied quite a bit. A guy named John Gherkin out of my office studied that morphology there, and it's pool riffle with, you know, there are pools and runs and pools and runs, but it's a small stream and it's not braided at all. And Inconnu and humpback whitefish are spawning there. It's all over the board as far as like what the habitat is that they use. But I think the important part is that you have gravel and you have water that's, you know, either percolating up or oxygenated that's bathing them, and how that happens, whether it's through these big braids and subsurface flow, or you know, being when they are spawning, when Inconnu have been spawning in those smaller systems, they spawn in the runs. That's where the eggs distribute. So there's flowing water over them all winter. But as far as changing a habitat that has been altered, say with gravel, you know, stream bed gravel mining or something like that, I don't have a clue how to go about doing that. But so I don't know. I've kind of talked around things, but I don't think I answered your question.
No, I appreciate that. I mean, that's we're just starting to learn more about that, but the thought about the diversity of habitats and what's important, that's certainly a helpful bit of advice. Thanks.
Yeah. Well, in a lot of these spawning areas, you'll find multiple species like that upper Yukon flats. You know, when we did the electroshocking for the population estimate there, there'd be places where 50 broad whitefish would come to the surface, you know, as you'd be going along, or, you know, 100 Inconnu would come to the surface, and Bering Cisco and humpback white. It was full of fish. And then there were burbot in there that were eating Bering Cisco specifically. They're just the right size for them. And there were pike in there, and there were fall chum salmon and coho salmon, and I don't know if they were spawning there or not. Lake chubs all over, probably eating eggs once they started spawning. But these habitats, they look like desolate landscapes as they get close to winter. They're absolutely packed with fish. So it's really kind of stunning.
Cool, thanks.
Yeah.
All right, thanks for that. Some good thoughts for folks in the Great Lakes. So we're coming up on an hour. So unless we have any other questions, we might wrap it up. Just to remind folks that we don't have September's speakers quite confirmed yet, so there'll probably be an email to follow up on that when we know. And then just as a reminder moving forward, that Erin Dunlop will be speaking in October on some of her work on lake whitefish recruitment and early life history. So, one more call for questions. Anybody going once, going twice? All right, well, thanks again very much, Randy.
Well, thanks for having me. This has really been an honor.
All right, well, have a good weekend, everybody.
Thanks, Randy. Take care.
Thank you.